Here we go. And should be putting subtitles at the bottom. Yes, there they are. Perfect. Okay, so before we go into all of this vaccine stuff, we got to make sure we're all on the same page with molecular biology. And I know that's not going to be the case, so here we go. This is this is how we do that. So first of all, this is going to be how proteins get made. And so the goal of a vaccine is going to be to do something that ends up producing protein and then using protein to provoke an immune response. And we'll have another one of these that goes through what an immune response looks like and how you make it. But for right now, let's just focus on the basics. So the way it works in normal biology is that you have DNA shown up here at the top, and DNA is going to get turned into RNA. And there are special enzymes that do that, and then RNA is going to get turned into protein, and a special enzyme called the ribosome is what does that. DNA can be copied to make more DNA, and so that's the general case, is that you get this flow of information from DNA to RNA into protein. And each of these is just a chain. I don't mean protein as in eating lean meat. I mean the physical molecule that's made of only amino acids, arranged in a chain that's then folded up into a three-dimensional shape. I'll show you more about what those look like. There's a separate little presentation for that. But we'll keep this simple. So this is the central dogma, is that this is the way information flows. So all the blue lines. But there are exceptions to everything, even the central dogma. Oh my goodness. And so there are cases where RNA can be copied to make more RNA. Doesn't normally happen, but things like coronaviruses do this all the time. There are cases where RNA can be turned back into DNA. Viruses like HIV are good at this. Coronaviruses don't do it, but some others do. And then, you can copy protein to make another protein in a few interesting ways. They're not particularly relevant to what we're going to be studying here, which is the coronavirus. So, all right, what do I mean by protein? What do I mean by amino acids? Well, there's 20 different amino acids, and they're shown over here. If you see down at the bottom, they all have this NH2 and COOH on the other side, except for one of them, where the NH2 is actually joined up to the COOH, and in joining it had to lose one of those hydrogens. So this is the one exception. But all the rest of these have the same bottom part and a different top part. So the bottom part gets joined together into a chain. We're actually going to join this COOH onto that NH2, and we'll take off a water molecule. So we'll lose an OH from this side and an H from this side, and boom. Once you put two amino acids together, you have now made a protein. And you can make your protein any particular length. And the properties and the structure of this protein are going to depend on what those side chains are. So there are a couple of common ways that the side chains can fold up, or rather the whole protein can fold up. So look in here, this little blue, gray, gray, blue, gray, gray, just like that. These little dots going around. This is going to be the protein backbone. So this is the part that is the same on every single amino acid. The side chains are going to stick out kind of sideways, and we haven't even shown the side chains on these molecules. We're just looking at the backbone here. There are a couple little tricks that the backbone can do. One of these is it can make something that looks like a coil. It looks like an old-fashioned telephone cord, and this is called an alpha helix. And so you can see your helix here. We're twisting around and around and around, only we're doing it in 3 dimensions. And an alpha helix is held together because one part of the backbone, so this is that oxygen that's sticking out over here in the COOH, it's the double bonded oxygen, is going to reach up and try to steal an electron from the nitrogen, which is the little blue one here. And oxygen trying to steal from nitrogen is what you call a hydrogen bond. There's actually a hydrogen involved here that's being kind of pulled, and we get sort of a nice interaction. And that is strong enough to hold the entire alpha helix together. So that's one thing that the backbone can do, is alpha helix. The next thing that it can do is beta strand. And in a beta strand, all of your backbones are going to be lined up nice and straight, and then there will be some kind of curve at the end, and then another nice straight part, and they're going to be held together. They're basically going to be stapled together in a flat line again, with interactions of the backbone. So that oxygen is going to be trying to steal this, the electrons from this hydrogen, and that is what a hydrogen bond is. You see these little things called R on the side. Think of R as the rest of the molecule. So the backbone is what we're seeing here, and the R groups are going to be different depending on whether you have this molecule or this molecule, just like that. And so, yeah, there will be certain amino acids of these 20 that are happy to be in an alpha helix. There are some that will be happier to be in a beta strand, and 2 or more beta strands makes a beta sheet, and so that's what you'll see. When we're talking about protein, we're going to be talking about it at this level, at the level of a particular kind of amino acid that's involved in a particular, what we call secondary structure, the alpha helix and the beta strand. The third type of structure is basically a garbage can that we put all the other structures into. We call these things random coils, or if they're bendy, we'll call them a flexible loop. And this is just literally anything that's not an alpha helix and not a beta strand will be called a random coil. So think of it as like the third thing. If you're A, B, C, it's A alpha, B beta, and C coil. And that's all it takes to make proteins. These structures are what makes up a protein. And then they'll be arranged in some three-dimensional configuration, and that'll be the overall shape of the protein, which is what we call a protein fold. So there are certain amino acids that are happier than others in certain types of secondary structure, like in that helix structure or that beta strand structure. So shown here, down at the bottom, we can abbreviate each of those 20 amino acids with a particular letter. And you don't have to memorize the letters for now, but if you already know them, this is great. You'll get to use that, and it's gonna make things easier for you, because you won't have to look certain things up. But whatever, this is a thing you can totally look up. So V is valine, I is isoleucine, L is leucine, M is methionine, etc. This part's easy. And so the propensity is showing you how much it prefers to be in an alpha helix. Like for, what's the ratio of methionines in an alpha helix to methionines not in an alpha helix? And so this is about, a little more likely, maybe 1.5 times as likely to be in an alpha helix, something like that. You can get any amino acid in any place. It's just that they have tendencies, and by knowing these tendencies, we can understand them. For example, look at valine down here. It would rather not be in an alpha helix, so it's below a propensity of 1, which means, no, it doesn't belong in a helix. But look at that. Beta strand propensity is about 2.0. So valine would really like to be in a beta strand, and it would very much prefer not to be in an alpha helix. There we go. That's the kind of level that gives you the understanding. And so you can actually go through a protein. You can line up little bits. So I've blown them up in the box, but at the top we've got little bits from the this protein here, and at the bottom we've got little bits from this protein. And so you can say, okay, if it's got M, A, L, E, or K, these are the number 1 alpha helix amino acids. You can just count them up. So like in the box, how many M, A, L, E's, and K's? So L, 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 K, L, L, that's a total of 6. And those are going to make it an alpha helix. T, V, W, I, F, and Y are going to make it more beta strand. And so there are none of those in this box. And so you can go through and you can end up predicting, okay, this one's going to be alpha. And looks like that got moved off a little bit. This one's going to be beta. This one is kind of even, so we're not really sure. But anyway, that's how you can look at these things and get some idea of what kind of protein shape they're going to make. And then, of course, you can solve the structure of the protein, or rather other people can, and they can figure out that, okay, this part makes an alpha helix, this part makes a beta strand, as does this part, this part, this part, and this part. Everything in the middle is some kind of random coil or flexible loop, and so they've called them L for loop, and alpha for alpha, and beta for beta. That's the B with a little tail on it, and the alpha looks like a little fish. So there we go. That's our little intro to protein structure. Now we're going to look at this in a different way in the next one.